an homeostatic organ. All right. Now, the liver is the second largest visceral organ. Of course, the largest organ is the kidney, and the second one to that is the liver. And this is found immediately below the diaphragm in the abdominal cavity. All right, so it's just, this is the thorax. Below the thorax, you have the diaphragm that separates the thorax from the abdomen. Immediately after that, you see the liver spanning from the right to, the left to the right. All right. Now, most of its functions are related to controlling metabolic activities essential for maintaining good blood composition. That's true. Now, its activities are so concerned with metabolism of food absorbed from the GIT, that's gastrointestinal tract. We get to see how how the liver helps to manage the content of food that comes in, how it screens it. Now, it has two major lobes, the right and left lobes, which will be seen in a moment. Let me see if I have that. Yes, I think I have that. So you can see it has right and left major lobes. Then the left lobe is further divided into caudate and quadrate lobes. All right. So those are, you can see that in that animation on the right here. So right and left lobes. This is the right lobe, the left lobe, which is the left lobe is smaller, and the right lobe is one that has caudate and quadrate lobe. Please, is the right and the left the right lobe, not the left lobe. That was a mistake. So please bear that in mind. So the one thing you need to know is that the liver has ability to regenerate. All right, that is one thing. I'm not sure I wrote in this thing. When a particular lobe of the liver is taken off or becomes diseased or something, it has a way of regenerating, and that's because it does a lot of function. So it is given ability to regenerate by itself to a large extent. Now it is covered with, it's not concerned, it's covered with tough fibrous capsule. All right. Now a unit of liver consists of different things, and that unit is called lobe, and it consists of hepatic artery, vein, then hepatic portal vein, then bile duct. That means there are four things there. Hepatic artery in a lobe. So that means the unit of liver is called lobe, and that has four components. Basically, the hepatic artery, a vein, as it were, then the hepatic vein and hepatic artery, I mean to say, then hepatic portal vein, then bile ducts. So those are the things you see in a lobe of liver. You get to see that as we move on. Now it contains cells called hepatocytes. So the cells of the liver are called hepatocytes, which can have prominent nuclei and Golgi apparatus and many mitochondria and lysosome. Now lysosome is present because they help in detoxification and they do a lot of things, and they have granules, glycogen and proteins, and droplets of fat, so to say. Please, not of these. I'm sorry for those typographical error. So let's move on. Now, the liver cell consists of special type of cell called Kupffer cells. Kupffer cells, which are actually macrophages with phagocytic ability. What do? They have ability to engulf bacteria and some other things that comes into the liver. So that's one of the ways the liver helps to fight against diseases, fight against and destroy things, so to say. So Kupffer cells are macrophages that can engulf phagocytes, of course. All right. So the food absorbed from the small intestine goes to liver via hepatic portal vein. All right. So you can see that here. So this is the small intestine here. So when the food from the duodenum and ileum has been digested, it goes straight into the liver by hepatic portal vein. You can see that here. You can see hepatic portal vein taken to the liver, and it's going to the liver because the liver has to screen that absorbed food before it goes into general circulation. All right. Now, I did talk about the lobule. This is a lobule of the liver that consists of hepatocytes, that consists of bile ducts, hepatic artery, hepatic vein, then hepatic portal vein. All right, so this is a lobule of the liver. And there's something I want to quickly explain here. You see something here called sinusoid. Sinusoid, yes. Sinusoid are just canals that connect central vein to the branch of hepatic portal vein. All right. Sinusoid connects hepatic portal vein and central vein together across. All right. That's just this actually from a advanced level to some extent, please. All right. So moving on, what are the functions of the liver? These are the functions of the liver. You can pause the video here to see that. All right. So carbohydrate metabolism. The liver receives digested food from small intestine via the hepatic portal vein. We saw that not quite long. Now the liver prevents fluctuation of the glucose, and this is achieved by one, converting all exotic sugar to glucose. Now because you know we have galactose, which is also a 6 carbon, so all of them are converted to glucose, all right, which is the universal sugar, so to say. Now the conversion, now what happens is that the body will use part of the glucose. Now the excess glucose will be converted to glycogen. So converting glucose to glycogen, which is glycogenesis, is done by the help of insulin from the pancreas, from beta cells of pancreas, which is islet of Langerhans. And this occurs in response to high glucose level in the blood, that's hyperglycemia. However, glycogen is also stored in the skeletal muscle. So it's not only stored in the liver, it's also stored in the what? Skeletal muscle. As we move on, you understand what happens to the glycogen stored in the liver is different from what happens to the glycogen stored in the skeletal muscle. Now, the stored glycogen is converted back to glucose by glucagon. When the sugar level drops in the body demands like a deposit and a withdrawal. Insulin has deposited glucose as glycogen in the liver. When the body is in need of energy, glycogen will go back there to withdraw part of the glycogen deposited earlier on to release it, change it to glucose so it can be energy, because our body cells relies majorly on glucose. That's why other food substances must be converted to glucose by a process called gluconeogenesis. If you have seen my video on respiration, anaerobic respiration and aerobic respiration, I talked about that there. Now the response is triggered by low glucose in the blood, that's hypoglycemia. Now the need for more glucose can arise from danger. Of course, when you are in fight or flight, you want to run, you need energy to run. In stress, in cold, then when you are fasting, then adrenaline and noradrenaline. Of course, you know these two hormones are secreted majorly in emergency, so they will help to convert glycogen back to glucose. All right. Now it is important to know that the conversion of glycogen to glucose is different in liver and skeletal muscle. Now the difference will be explained in a minute. I think I said that now. Now the conversion of, there's also, yes, what also the liver also does in some orchestration is conversion of non-carbohydrate substances to glycogen. That's gluconeogenesis, like conversion of excess protein to glucose. Now amazingly, some excess glucose, that's funny, after you have all the glycogen house has been filled up in the liver and in the skeletal muscle, the excess glucose left afterwards is converted to liver to fat also. Of course, you know that can also be converted back to glucose later on when there's a need for that. Now, looking at this diagram here, you see a balance of glucose in the blood. That's about 90 milligram per centimeter per 100 ml of blood. All right. Now, when around here, we start around here. When there is, when you are just finished eating a nice food, let's say it is written here 4 Jolly Donuts. We'll zoom that in. Now, yes, the glucose will rise. When the glucose level rises, that stimulus will go to what? You see elevated blood sugar level will go to pancreas to secrete what? To secrete insulin. Insulin will do what? Will convert, look at the liver here, convert glucose to glycogen. Then uptake of glucose will be increased by cells. It will also tell cells to absorb more more glucose. That is insulin doing that instruction. Then later on, when blood glucose level, when that has happened, after storing glycogen in liver cells and body cells, skeletal muscle and the likes, and increasing the absorption, the blood glucose level will fall to homeostatic set point. So stimulus for insulin will be diminished. There's no need for insulin. However, when blood glucose level drops after skipping a meal, while fasting, need for you to run, what happens? There's low blood sugar level, that's hypoglycemia. It will stimulate glucagon to be released from the pancreas. Then, like I told you, it's like a deposit and a withdrawal. Then glucagon will go to the liver to convert glycogen to glucose. Then the glucose is released straight into the system to balance up. You can see, so blood glucose level rises, homeostatic set point is achieved again. That balance is achieved again. All right. Now, looking at this here, this is glycogen converted to glucose phosphate, and all that is released. The glycogen stored in the liver, when it is broken down, is released for general circulation. All cells of the body will use it. But the glycogen stored in the muscle cell is only for itself. So when the glycogen is broken down, it changes to glucose phosphate, then it changes to form what's it called? Pyruvate, which will lead to formation of glycolysis, which is formation of ATP, energy. So energy will be, that's why you saw that energy is released here. So the glucose formed is broken down by glycolysis, then ATP is formed. If you have seen my video on respiration, so ATP is formed and that is for the skeletal muscles itself. So the glycogen is for skeletal muscle itself. So when that is formed, the pyruvate that is formed can either go back to form glucose or glycogen, or you can actually go further to enter Krebs cycle and the likes. So please bear this in mind, that the glycogen broken down in the skeletal muscle is for itself when it needs that, when we need sudden energy. All right, so it's quickly broken down. But the one from liver is for all the cells of the body. All right. Now, protein metabolism. The fate of protein are for growth, repair of cells in the body, and other aspects of metabolism. But protein is not stored. That's funny. So protein must be stored in different forms. So the liver handles the protein in the following ways. It uses to form plasma protein synthesis. It uses to form plasma protein. Deamination, which is the removal of amino group from amino acid to form ammonia. So deamination is actually the removal of amino group from amino acid to form ammonia. Then urea formation. So that is the conversion of ammonia, because ammonia is toxic, to urea, which will be excreted by the kidney. All right. So bear that in mind. So it's in sequence. When we deaminate amino acid, we convert it to form ammonia. Ammonia is then used to form what? Urea also. Then transamination, which means the transfer of amino group from amino acid. It's not all of the amino acid that will be deaminated. So some of them will just be transferring of that amino acid, so the amino group, to other organic molecules, basically. So also the liver helps to store minerals. It helps to store minerals, both the soluble. I'm sorry for this typographical error. Please, I'm sorry for that. Now it's for both for the soluble and both for the fat soluble. You can see that they're supposed to be fat. I don't know that what changed that. So fat soluble vitamin, which is the ADEC, and water soluble vitamin, which is at the B and C. All right. Now the liver also helps to store microelements such as copper, zinc, cobalt, molybdenum, and some ions too. All right. So that's what the liver helps us to store. Then bile production. Now, from formation of, while the liver is carrying out its metabolic processes, bile is produced by the hepatocyte and is stored and concentrated in what? Gallbladder. So in case you are asked, what forms bile? It is the liver. But where is it stored? In the gallbladder. So the bile is composed, it's a greenish substance actually. It's composed of 98% water, 0.8% of bile salt, 0.02% of bile pigment, 0.7% of inorganic salt, and 0.6% of cholesterol. Now this is very fun. Like I said, liver uses cholesterol, or forms cholesterol as a content of bile. And bile cholesterol is very bile is very important in the formation or digestion of fat. So the bile helps in the emulsification of fat in the duodenum. Without emulsification of fat will not be digested. All right. And I used to say that cholesterol should not be too much, but it's also essential because it's part of the content or constituent of bile that helps emulsification of fat. So it means if you don't have any cholesterol at all, there will be issues with fat digestion because it's part of bile. All right. So you can see that here. So this is the gallbladder. This is gallbladder here. Then it's emptied into duodenum where it's emulsified fat coming from the stomach to the duodenum. All right. So this is an animation to show that you can see content of stomach move to duodenum. You can see the yellow thing coming from the liver to the gallbladder. Gallbladder going that's the bile from to the sperm. Through the bile duct. So the bile duct takes it from gallbladder to the duodenum. You can see that there. That's where it's been emulsified. All right. Now, storage of blood. The blood vessel coming from spleen and gut join to form hepatic portal vein, which together with some other blood vessel carry large blood vessel. So the blood to the liver. So the liver helps to store large amount of blood to some extent. In fact, it receives a very large amount of volume of blood compared to what's stored to the supplied to the entire body. All right. Now, formation of RBC. Now, during intrauterine life, when we were in our mother's womb, the liver of the fetus helps in the formation of red blood cells. Now, it does this not all the way. Initially, around the first trimester, the first 3 months, it is the yolk sac that forms red blood cells. Around the 6th month, the spleen and the liver takes charge. However, later in development, the bone marrow, that's last trimester, that's the last from 7, 8, 9 months. The bone marrows of the bone of the child becomes the site for production of red blood cells. However, the liver then becomes the one that will destroy red blood cells later on. All right. Now, breaking down of hemoglobin. Now, red blood cells take about 120 days to live. That's about 4 months approximately. Now, when these old red blood cells are old to some extent, they are destroyed by phagocytic cells of the liver. That's your Kupffer cell we mentioned earlier on, and spleen also. Now, in the process of breaking down red blood cells, because red blood cell has hemoglobin attached to it, so hemoglobin is broken down to release heme and globin. Globin is broken down to amino acid, and that amino acid is reused, while the heme, which is the now the heme has iron parts and the non-iron parts. So the heme has the non-iron part of heme, which is then broken down to form biliverdin, which is green. Then that's wanted to call bilirubin. So just know that bilirubin is formed from the breaking down of hemoglobin. All right, I'm just breaking it down because globin is the amino acid or the protein part of the hemoglobin. Heme has non-iron part and iron parts. The iron part is also reused, but the non-iron part is broken down to form biliverdin, which is later used to form what? Bilirubin. And that yellow bilirubin is what gives our urine some sort of yellowish amber color. All right. Now, detoxification. Now, detoxification is the process whereby the liver helps to neutralize toxins and poison consumed. Now, when I say detoxification, it doesn't really mean it changes what was toxic to non-toxic. No, what it does is that it sieves. The liver will hold on to the toxic aspect of what is consumed, then allow the non-toxic one to pass so that it doesn't harm us. So that's how it does it. So the actual process which it used to do that is reduction, oxidation, and methylation of different substances that comes into the body. So bear that in mind that it helps to, it screens the thing, it holds on to it. Sometimes, to some extent, it neutralizes some things, but it doesn't neutralize all things. All right. Then inactivation of hormones. Now, liver helps to destroy hormones after their effect has been executed. Yes. For example, adrenaline after use. When adrenaline has been used, for example, as we'll be seeing here, see what happens to this young lady here. That guy attached a string to what was on the floor. Then when the girl saw it, she thought it was a snake. At that instance, adrenaline is released. That was what made her to run that way. After some time, if this video is seen to the was shown to the end, the girl will know it's a prank. And if it's a prank, the whole thing that has happened, like now she will so run as she's running, that adrenaline was what brought about that straight, that running, that energy, that breathing and all that. After that, the liver will, once she knows it's a prank, that it's not a snake as it were, or if it was a snake and she escaped it, the liver will cancel out the effect of adrenaline because it will be absorbed if she keeps running, even after she knows it's not a snake or if she has escaped being killed of the snake. All right. Then heat production during extreme cold conditions with the influence of adrenaline and thyroxine, the energy releasing activity of the liver increases. And you know, once you release energy, the more the heat is produced. All right. So let's move on to the liver diseases. We have liver cirrhosis, hepatitis, gallstone, liver abscesses, then jaundice. All right. Now, the liver cirrhosis, this is when the parenchymal cells of the liver are destroyed and are replaced by fibrous tissue, which eventually contract blood vessels, leading to decreased blood flow to the liver. So the liver becomes turgid, and all the blood vessels passing through that are squeezed. At the end of the day, the liver becomes turgid and hard. Then, as time goes on, the liver cell begins to die, and this is caused by chronic consumption of alcohol over a long period of time. Accumulation of fats. That fat can actually squeeze the liver cells, and when you squeeze the liver cell, blood flow reduces and the liver cells will end up dying. All right. Then we have injection of poison such as carbon tetrachloride, viral disease such as infectious hepatitis. All right. So those are the causes of liver cirrhosis. Then this is how the liver will look like. You see the normal liver, then see what the liver becomes after it has become destroyed by these various factors. Then we have gallstone. This is the abnormal condition in which the cholesterol in the gallbladder precipitates. You know, we told you that, I said at the beginning, that one of the contents or constituents of bile is gallbladder. So the amount of cholesterol is dependent on the amount of fat consumed. So if you consume too much of what fat, there's tendency that there can be too much of cholesterol. There will be too much of cholesterol, and this can lead to, now when there's too much absorption of water from the bile, you know, you have sucked out water, so what you have left is just the bile salt and the what? Cholesterol that will precipitate. All right. Then too much absorption of bile acid from the bile also can also lead to gallstone. And too much of cholesterol in bile, like I said, if you eat too much of fat, the inflammation of the epithelium of the gallbladder. Now, when the epithelium of the gallbladder becomes inflamed, absorption in and out becomes affected, and that can lead to precipitation of the cholesterol. All right. So this is what it looks like. You can see the stony masses here. Those are bile. Those are gallstones. All right. You can see it here. There's a blockade that will prevent the flow of bile out of the gallbladder when it is needed. All right. Then we move on to liver abscesses. This is a collection of rubbish fluid which is pus within the liver. This can be caused by a protozoa called Entamoeba histolytica. It's one of those what's it called? Protozoa. It helps to, it's a protist. When it releases toxin into the person's body, it causes, can you see that the liver looks normal on the outside, but within there is a collection of dead tissue and fluid, pus within that. All right, that can be cured by going to the hospital, then you're giving a drug. Sometimes it can be drained. It can be, depending on how severe it is. All right, so it can be done surgically. It can be an antibiotic can be given to kill the pathogen, which is the amoeba in this case. Then the next one is jaundice, or jaundice as it's called. And this is yellowish coloration of the tissues of the body due to abnormal increase in level of bilirubin. I'm sorry for this. I think I have a lot of typographical error here, but I'm here to correct it. Bilirubin, there's no O after the N. All right. Now this yellow coloration is more noticeable in the white of the eye, which is the sclera of the eye, and other mucous membrane like nail bed, because the eyes is white, that sclera is white, so you easily notice it there. Can you see this young child here? Can you see? Yes, he has jaundice. You can see he has bilirubin. I have bilirubin in my body, but because it's not too much, it won't show where it ought not to. So you can see on the palm of this old man, that's the normal one. The normal one, you can see yellowish coloration because it's fair, so it shows more. All right. In fact, on the skin of a fair person, can actually see that yellowish coloration. Is that clear? Now you can pause the video here to just look at the various causes of jaundice. Now one of them is, let me zoom this out so you can see that. Now you can see the causes. The fourth one is hemolytic anemia. The red blood cells will be destroyed, ought to be destroyed every 120 days. And anytime red blood cell is destroyed, hemoglobin is destroyed. And when hemoglobin is destroyed, we form bilirubin. So when you have hemolytic anemia, as we've seen in sickle cell anemia, the person's red blood cells die earlier than 120 days. So when this happens, it causes too much of bilirubin to be formed. And when there's too much of bilirubin, it will lead to what? Yellow coloration of different parts of the body. And also it can be obstruction of the bile duct. Now hemoglobin is dying normally, red blood cell is dying normally, it's not too much, bilirubin is formed, but because it cannot find its way to the gallbladder, it has been obstructed. That's obstructive bile duct, or they call obstructive jaundice. That one it will cause, once it cannot pass to enter the gallbladder, it will flow back into circulation, that we see it in different parts. So those are other causes of jaundice. Now we move on to hepatitis. This refers to liver damage, which can be caused by many agents. So hepatitis simply means infection or swelling or damage to the liver. So it's not specific. So it's characterized by what? Swelling and inadequate functioning of the liver. It can be caused by any of the following viral infections. So you have hepatitis B, C, and the likes. Those are different types of viral hepatitis. Then we have excessive alcoholic consumption, poison, excessive administration of some drugs, e.g. paracetamol. So there are lots. So hepatitis should not be seen as viral hepatitis, which is the most common one that people that we take vaccines for. But it is caused by a lot of diseases. All right. So in summary, this is just different stages of liver damage. Thank you very much for being with me on the lecture. Do have a wonderful